There are three stages that a cell can undergo with the signal transduction pathway, which are reception, transduction, and response. For liver cell, epinephrine, or adrenaline, will be the signaling molecule for the G protein receptor system. The G protein receptor is attached to the plasma membrane, which consists of a phospholipid bilayer that only allows nonpolar hydrophobic molecules to go through, and guanine diphosphate is bonded to the G protein that is inactive. Guanine diphosphate is dephosphorylated from guanine triphosphate, where one phosphate group and a lot of energy is released. When the signaling molecule, or epinephrine, comes in contact with the G protein receptor, this allows for the guanine diphosphate that is attached to the inactive G protein to activate and send the signals on a cascade or will go through transduction. As it goes through transduction, the signal will be sent to multiple enzymes where they will decide to either act upon it or halt the system by terminating the pathway of the signal and go through apoptosis. For this scenario, epinephrine will typically go from the G protein to adenylate cyclase, which will convert ATP to cyclic adenine monophosphate and activate the phosphorylation cascade, where it will activate the protein kinases. Lastly, the cell will respond by releasing the glucose into your bloodstream and going through fatty acid synthesis.
In the beginning of the tyrosine kinase receptor system, there are two receptor tyrosine kinase proteins, the ones in blue. The ligand binding site is on top of the cell membrane, while the three semicircles on the bottom are tyrosines. The ligand binding site awaits a signal. In this case, a ligand, here in green with a yellow center, slowly approaches the binding site on each receptor. Once it attaches, the two tyrosine kinase proteins bind to each other, forming a dimer. This results in the activation of the tyrosines in the receptor. After that, 6 ATPs convert to 6 ADPs, which attach to the ends of each tyrosine, fully activating the receptor tyrosine kinase. From the inside of the cell, several inactive relay proteins approach all the tyrosines. The relay proteins get activated and carry a cellular response deeper inside the cell. This ends up triggering multiple transduction pathways at once. After that, the cell regulates the cellular response through the discharge of calcium ions from the ER.
Down syndrome is caused by an extra copy of chromosome 21. Its symptoms are delayed speech development, lack of muscle tone, congenital heart disease, and a distinct facial structure. Treatments include speech and physical therapies as well as special education. Since there's an extra copy of chromosome 21, the Down syndrome critical region DSCR2 gene is expressed more often than it should be. The gene controls calcium release in cellular communication and causes less calcium ions to be released. Another gene that is overexpressed is the tyrosine kinase gene that inactivates NFAT, which is a transcription factor that activates genes that code for proteins that function in cellular communication. Cells without Down syndrome will send a chemical signal to another cell who will receive, transduct, and respond to that signal, but cells with Down syndrome cannot respond accurately to signals that they receive.